Let's dive right in, no messing around. Homeostasis is basically your body's ultimate balancing act. It is the regulation of your internal conditions. Think things like blood glucose concentration, temperature, and water levels, keeping them perfectly stable even when external conditions are going absolutely crazy. So why do we actually care about this? Well, because this stability is what keeps crucial chemical reactions involving enzymes ticking along at their optimum rates. And if you want to secure full marks on the paper, remember to always use that specific keyword, optimum.
Now, this kind of balance is incredibly critical when your body has to react to danger. The reflex arc is all about one thing: speed. The signal pathway goes like this: stimulus, receptor, sensory neuron, relay neuron across a tiny gap called a synapse, then to the motor neuron, and finally to the effector. Notice what's missing from that list? The conscious brain. The signal actually bypasses your brain entirely and loops right through the spine so you can react instantly. A classic exam question here involves testing reaction times by dropping a ruler. And here is a pro tip to secure a top-tier 6 marker. Mention that you can calculate the exact reaction time from the distance the ruler falls using the physics formula S equals a half AT squared. They love seeing that cross-subject knowledge. All right, triple science alert. If you're studying foundation or double science, just sit tight for a second. For you triple students, you absolutely need to know the brain inside out. The cerebral cortex, right at the top, handles all your higher-level functions, stuff like memory, speech, and problem solving. Then you've got the cerebellum at the back, which is all about motor skills, balance, and coordination. And the medulla, that little part controls your unconscious actions, like your heartbeat and your breathing. Basically, the things you fortunately don't even have to think about to stay alive.
We are still strictly in triple science territory here. Let's look at a brilliant comparison regarding the eye. Myopia is short-sightedness, meaning you simply cannot focus on far objects. Hyperopia is long-sightedness, making near objects an absolute blur. Now, to actually focus, your eye uses a process called accommodation. For far objects, your ciliary muscles relax, the suspensory ligaments pull tight, and the lens gets thinner so it refracts light less. But for near objects, the exact opposite happens. The ciliary muscles contract, the ligaments go slack, and the lens becomes fatter to refract light much more heavily. Get that mechanism straight in your head for the exam.
Okay, pulling everyone back in now. Double and triple. We're jumping from the lightning-fast nervous system to the much slower, chemical-based endocrine system. This is an entire system of glands that secrete hormones directly into your blood to reach specific effectors. The big boss of this whole setup is the pituitary gland in your brain. It's known as the master gland because it secretes hormones that literally just tell other glands what to do. You've also got the thyroid controlling your metabolism, the adrenal glands handling your fight-or-flight response, and the pancreas managing your blood sugar. Speaking of the pancreas, it uses two specific hormones to manage that blood sugar: insulin to lower it and glucagon to raise it. But what happens when this delicate system goes wrong? You absolutely must not mix these up on the exam. Type 1 diabetes is when your pancreas simply cannot produce enough insulin, meaning these patients need regular injections. Type 2 diabetes, on the other hand, is when your body's cells basically stop responding to the insulin you actually do make. Type 2 is heavily linked to obesity and is usually managed by changing your diet. Time for another triple science alert. Let's talk about kidneys. Your kidneys constantly filter your blood, keeping the perfect water balance while getting rid of toxic urea. This balance is controlled by a hormone called ADH, which comes from the pituitary gland and utilizes something called negative feedback. So if you're dehydrated, more ADH is released, causing the kidney tubules to reabsorb more water back into your bloodstream. If your kidneys fail entirely, a dialysis machine has to step in to physically filter that dangerous urea out of your blood instead. Bringing everyone back to the core syllabus now. The menstrual cycle is driven by 4 key hormones, and you need to know their exact chronological order to guarantee full marks. First up, FSH from the pituitary causes an egg to mature. This then triggers estrogen, which thickens the uterus lining and cleverly shuts down the production of any more FSH. Then LH steps in to trigger the actual release of the egg. Finally, progesterone steps up to maintain that thick uterus lining, keeping the body prepped for a potential pregnancy. Now, if natural reproduction isn't an option, medical science can step in with IVF, or in vitro fertilization. First, the mother is given injections of FSH and LH to induce egg maturation and release. Then, here's your exact exam checklist. Step 1, harvest the eggs. Step 2, fertilize them with sperm in a lab. Step 3, let them develop into embryos. And Step 4, insert them safely back into the uterus. But to grab those crucial evaluation marks, you must remember to mention the drawbacks. IVF has a very low success rate, it is highly expensive, and it carries a significant risk of multiple births. Foundation and double students, you get to skip this next bit. Triple science alert. Plants use hormones just like we do, and the exam board wants you to know 3 of them. Gibberellins give seeds a vital wake-up call to start germinating. Ethany is the gas that induces the ripening of fruits. And auxins, they are the absolute stars of the show. They control phototropism, which is bending shoots towards light, and geotropism, making roots grow downwards with gravity. Now here's the kicker. Auxins gather on the shaded side of a shoot, causing an unequal distribution, which leads to cell elongation on that side. You need to learn that exact phrasing, unequal distribution. All right, everyone, back in. And this right here perfectly illustrates the single most vital difference you will face in the genetic section, mitosis versus meiosis. Mitosis is strictly for asexual reproduction and cellular growth. It divides a cell to create 2 genetically identical clones. Simple as that. Meiosis, on the other hand, is exclusively for sexual reproduction. It takes place in the testes or ovaries and goes through 2 divisions to create 4 unique haploid gametes, like sperm and egg cells. This means meiosis is entirely responsible for introducing that all-important genetic variation into the offspring. Let's zoom right in on the actual code of life. Your genome is the entire set of genetic material you possess. Fun fact, this was fully mapped out by the Human Genome Project back in 2003. DNA itself is a polymer shaped like a double helix, and it's securely held together by nucleotide base pairs. A always pairs with T, and C always pairs with G. Every sequence of 3 of these bases codes for a specific amino acid to build proteins. But what's genuinely fascinating is epigenetics. This is the study of non-coding DNA, stuff that doesn't actually make proteins, but somehow acts like switches, influencing how your other genes are expressed. Now, because we understand alleles, we can mathematically predict biological traits. By crossing 2 heterozygous parents in a Punnett square, meaning they both have one dominant big B and one recessive little b allele, we reveal exactly a 25% chance of the offspring inheriting the little b recessive trait. You use this exact same grid logic for exam questions on inheriting recessive disorders, like cystic fibrosis. Just map it out, and the marks are yours. And if the exam asks you to mathematically prove the probability of having a boy or a girl, doing this specific Punnett square is your guaranteed ticket to full marks. You put the mother's XX chromosomes on the top and the father's XY chromosomes on the side. Run the cross and you get 2 XX results and 2 XY results. That perfectly mathematically demonstrates the 50-50 probability of inheriting XX female or XY male chromosomes. Draw it exactly like that on the day. Let's look at the absolute titans of evolution. Charles Darwin championed random variation and the survival of the fittest. Jean-Baptiste Lamarck, meanwhile, argued that adaptation was guided by an organism's environment during its lifetime and then passed down. Now, to score highly on this explainer topic, use bacterial resistance as your ultimate proof of Darwinian evolution. When bacteria randomly mutate, the highly resistant ones survive the antibiotics and multiply. This is exactly why you are always told to complete your full course of antibiotics, to kill off the weaker ones and then completely wipe out the surviving mutated superbugs.
Of course, we don't really have to wait for evolution anymore. We have genetic engineering. Here is the precise step-by-step process you need to memorize for the paper. Step 1, cut the desired gene out using enzymes. Step 2, insert it into a vector, something like a bacterial plasmid or a virus. Step 3, insert that vector into the target organism at its early embryo stage so that every single cell develops with a new gene. Perfect examples to drop in the exam include genetically modifying bacteria to harvest human insulin for diabetics or creating golden rice that is packed with vitamin A. Triple science alert. Let's talk about cloning. Cloning plants is actually remarkably straightforward. You can literally just take a cutting and plant it, or use tissue cultures to mass-produce plants and preserve rare species from extinction. But cloning animals? That is infinitely more complex. One method you need to know is embryo splaying. You take a developing embryo, split the cells apart just after fertilization, and then put those identical cells into multiple surrogate mothers to grow into identical twins. But if you want to clone an animal that is already fully grown, you have to use adult cell cloning. Step 1, remove the nucleus from an adult cell, like a skin cell. Step 2, insert that nucleus into an empty, unfertilized egg cell. Now, the crucial point for the exam is right here at step 3. You must give that egg an electric shock. That shock is what jumpstarts the whole division process. Finally, step 4, you insert it into a surrogate mother's womb to develop normally. Moving on to the grand organization of life on Earth. Everyone is back in for this section. Carl Linnaeus created a massive classification system to organize literally all living things: domain, kingdom, phylum, class, order, family, genus, and species.
Need to memorize that order fast? Just use the mnemonic. King Philip came over for good soup. Easy. And don't forget the binomial naming system. An organism's scientific Latin name is simply its genus combined with its species.
Now, out in an ecosystem, absolutely everything interacts, and the exam wants you to categorize these interactions flawlessly. Abiotic factors are all the non-living elements: light intensity, temperature, soil pH, CO2 levels, and moisture. Biotic factors are the living elements: the availability of food, new predators, and pathogens. And if a question asks you to estimate how these factors affect population distribution out in the real world, remember to mention using quadrats for sampling populations and transects to measure how things change over a distance.
Every single ecosystem is powered by a food chain, which maps the flow of biomass and energy. It all begins at trophic level 1 with producers, usually plants or algae that use the sun's light to create biomass. Level 2 holds the primary consumers, your herbivores. Level 3 brings the secondary consumers, the predators. And right at the very top of the chain are the tertiary or apex consumers. These are the ultimate predators with absolutely no natural enemies hunting them. Last triple science alert of the explainer. Triple students, you must know how to visually represent these trophic levels using a pyramid of biomass. You might even be asked to physically draw one on graph paper. Always ensure it looks like a proper, symmetrical pyramid, not just a staircase. And if you are asked to calculate the percentage of biomass absorbed by the next level, just use the golden math rule. Take the bit, divide it by the lot, and multiply by 100. You can't go wrong.
Still on triple content here, why does a pyramid of biomass actually get smaller as you go up? Well, because biomass is constantly lost at every single trophic level. Animals burn energy simply to move around. They excrete waste like urea and water. They release massive amounts of CO2 through respiration. This continuous, unavoidable loss of energy and mass at each stage is exactly why food chains just cannot sustain endless trophic levels. Eventually, the energy simply runs out.
All right, everyone, back together for the final stretch. All of this biology we've just covered ties directly into human survival and global food security. Right now, food is becoming scarce. We have an exponentially increasing world population, changing dietary demands, and extreme farming costs. Plus, global conflicts, like the war in Ukraine, disrupt vast global wheat supplies. To survive, we desperately need biological, sustainable solutions. Sometimes, it's as simple as using fishing nets with larger holes so the young fish can escape, grow up, and continue to breed.
Which brings us to a provocative final thought that ties this entire explainer together. Could genetic engineering and targeted cloning ultimately be the key to solving our global food security and biodiversity crises? The biology you've just rapidly revised isn't just for passing an exam. It is the fundamental foundation for solving the world's most critical challenges. You are fully equipped with the knowledge now. Go into that paper 2 exam, trust your structured revision, and absolutely dominate those questions.